Mauro Costa Mattioli. So I first met Mauro when he was a postdoc in the Sonnenberg lab, and I looked up, and that's like almost 17 or 18 years ago, which is really scary. He had just begun his work on the role of EF2 alpha phosphorylation in memory, which is a truly innovative and unexpected avenue of research, but it was actually kind of a rocky start for Mauro and I, because he had just stolen a co-supervised graduate student that I had in the Kai lab. And instead of working on a shared project with me, the student was now just working on Mauro's project. I think by the time Mauro left McGill, he had 4 separate labs, all devoted to working on his project. So when I wrote letters for his job application, I said Mauro was a force of nature. So as most of you know, I'm usually wrong, but in this case, I think I was right, because in his own lab, Mauro has continued doing incredibly innovative and productive work, and he is truly a force of nature. He's continued his work on the role of the integrated stress response in neurobiology. He's shown important roles in many physiological and pathological processes, including Down syndrome, addiction, and traumatic brain injury. He's also made great strides in other areas. We're working on translational control in the nervous system, the role of mTORC2, the intersection between neurobiology and immunology, showing how translational control of immune regulators is really important for PKR and UPF2 and understanding the effects of mutations in these proteins on brain function. He's recently made huge strides in understanding the gut-brain interactions, and some of that's not even translational control. He's a recent recipient of the UCSF Presidential Chair Award. He's given a number of distinguished lectures, and I'm sure we're about to hear another one of them. So please, Mauro, take it away. Mauro, I don't hear you yet, so make sure you unmute yourself.
Okay, sorry about that. Can you help me with the pointer? How do you have a pointer in here? Oh my God. Anyhow.
The mouse usually works if you go into PowerPoint.
Okay, perfect. Sounds good. Okay. So first, let me start by thanking Wayne for such a very gracious introduction. I was not expecting anything less from a friend. And when I also thanks the organizer, Rachel, Graham, and Nahum for the invitation. This is one of the meetings that I was actually more looking forward, because it's been many, many years that I haven't seen, many colleagues and people that actually have inspired my work. And I always enjoy, having drinks at the bar at Cold Spring Harbor. So hopefully next year, or in 2 years, I will be able to come again and see you in person. So what I will tell you today is a story that started back in 2004, and this is about memory, which eventually is essential not only for animal survival, but it's also at the center of our identities. And at the time when we decided to focus on this, major focus in neuroscience was trying to identify specific brain regions that were important for memory formation and circuits that essentially could lead to different internal states. So obviously I am a molecular biologist. I am interested in the molecules which are required for a memory formation. I'm interested in knowing whether those molecules are hijacked in cognitive dysfunction, and more importantly, whether we can reverse those changes and then perhaps improve memory in some of those conditions. So there are 2 memory systems. This is the great German Ebbinghaus, psychologist who studied himself. This is a story of an N-on-1, but he came with 2 key principles for memory formation. One of them is that we have memories which are short-lived, short-term memory, and others which are persist for years, weeks, and even a lifetime. But those are the ones which are long-lived or long-term memories. And the other key factor that he discovered is that repetition make memory last longer. So it is practice that make it perfect. Now, the first empirical evidence that there were two memory systems originated in part by McGill and the characterization of a patient which is called HAIM. This is a patient that have seizures, and to remove or to alleviate seizures, a particular brain area was removed, the hippocampus and surrounding areas, and as a consequence, this patient was able to form a short-term memory but could no longer convert it into a long-term one. This is about 1957. The beginning of the 1960s, the first molecular distinction that short-term memory and long-term memory were different processes emerged by the study of protein synthesis and the addition of protein synthesis inhibitors, first by Agranov in goldfish in 1964, in 1965 by Sam Barondes in rodents, and later in the 80s by Eric and Dell in aplysia. They have shown that essentially to convert short-term memory into a long-term memory, you need protein synthesis. Not the young folks, but the old folks. I know that I was in Nahum's lab, and I was very interested in physiology. You know, at the time when I joined Nahum, most of the experiments were done in vitro, and I wanted to see whether whatever we were studying could translate in vivo. Nahum tried to convince me to study, of course, EIF4E and 4EVP, but I decided to focus on EIF2, and together with Nahum, they discovered that EIF2 is the factor which decides whether a short-term memory is converted into a long-term one. If we decrease the phosphorylation of EIF2, we increase the ability of the cell or the neurons to produce new proteins, and as a consequence, we are able to enhance long-term memory. And the opposite is also true. If we were to increase the phosphorylation of EIF2, now we inhibit the synthesis of proteins, and now we prevent the ability to form a long-term memory. So many investigators have reproduced and greatly expanded our work, most notably, probably as you have heard in the last translation meeting, my colleague Peter Walter, in a completely unbiased approach, discovered this fantastic molecule that essentially what it does, it inhibits the effects which are mediated by EIF2, and as a consequence, mimics the decrease in EIF2 alpha phosphorylation that we discovered genetically, namely is able to enhance long-term memory. And this has impacted not only the neurobiology of learning, but also industry efforts that now they are focusing in this pathway, as you will see in the next slide, which is the integrated stress response as a way to promote brain health in different conditions. So EIF2, as you know, is at the center of the so-called integrated stress response. There are 4 kinases, dedicated kinases that phosphorylate one site, serine 51 in EIF2. When EIF2 is phosphorylated, there is an inhibition of EIF2B, and as a consequence, translation is inhibited, but translation of specific mRNAs goes up. So this is not a simple switch for translation, but it's sort of a translation reprogramming. We have not only 4 kinases, but 2 EIF2 alpha, 2 phosphatase complexes that essentially regulate the phosphorylation of EIF2 alpha. And there's a lot of people that in here I should mention that they have contributed to this cartoon. Those are my favorite 3 individuals. Based on their work, I think we spoke with some of them, several times, but that really inspired my work, and they are sort of the fathers of the so-called integrated stress response. As you know, David discovered PERC, some of the phosphatase and the regulation, and Alan and Tom. I mean, those studies, in East, they were amazing. That they remain the test of time. The nomenclature is still difficult, but from the moment that I was able to understand the East nomenclature, I think were the basis for many of the studies that were later done in mammalian cells. Now, this is some of the evidence that the ISR enhanced long-term memory formation, and I will just describe one task in which animals will associate a particular context with a fearful experience, you give them a food shock. 24 hours later, you put the animals and you could measure their memory performance. And if you were to mutate serine 51 by alanine in the heterozygous animals, the animals in black, they have an enhanced memory. If you were to eliminate one of the EIF2 alpha kinase, you know, the ISR sensor PKR, either genetically or pharmacologically, you can enhance memory. And as advertised before, I mentioned, if you were to give ISRIP, that as we know from the work from Peter and David, binds to EIF2B and now promotes EIF2B activity, is able also to enhance memory formation. And again, there's a large number of people that have greatly reproduced and expanded our work, not only in normal memory settings, but also in pathological learning. This is an amazing paper that is still in the bioarchives in 2017 from the Dawson lab from Hopkins, in which what they do is they took a way to express the ribosomal protein in specific cells in the brain. And after this, they digested and they did a ribosome profiling. So this is just specifically in neurons in a particular brain region that is required for memory formation. If you were to train the animals in this particular case, consistent with what we discovered before, the phosphorylation of EIF2 alpha is decreased. And when they look at the translational program resembles to the one in which the phosphorylation of EIF2 alpha is inhibited. In other words, learning triggers a translational program that mirrors, in a completely unbiased approach, mirrors the inhibition of the ISR, or at least its major component. This is a paper which is coming out. One of them just came out from Eric Klan, another one which is coming out from Nahum's lab. In the brain, we have different cell types, and those different cell types, they can contribute differently to behavior. And Nahum and Eric, they have inhibited the ISR in this particular case in glutamatergic neurons. And when they inhibit the ISR in the specific cell types, they also see a memory enhancement. I have to say that I don't have any recollection from the field that a molecule works the same way that EIF2 does. There were many promises before, but I think that now you were to touch the ISR either upstream or downstream, and invariably you lead to an improvement in memory formation, something that when we originally thought to work on this was completely unexpected. This just not happened in rodents, also happened in chicks. And I gave a talk, Albert Einstein, many years ago, and I was able to inspire some of the students, and this is Jose Luis Peña and his student Gervasio Batista, decided to look in a model in which memory is impaired, and this is imprinting. So those are chicks that they will imprint into their mom, but the imprinting process, this is a learned and innate behavior. The imprinting process will take only the first 3 days. By day 4, if they haven't encountered mom, they're not gonna follow mom. And actually, yes, at day 4, they could follow mom, but the only way that they can follow mom is they will inhibit the ISR, either by using an inhibitor of PKR or treating the animals with ISRIP, demonstrating that this must be, is an evolutionary conserved process. Now, that gave us a lot of hope, because in conditions in which memory is impaired or memory cannot be formed, by inhibiting the ISR, we can bring memory back to life. And that's where we decided to look in pathological conditions. And Ping-Chun Zhu in my lab decided to work in Down syndrome, which is the most frequent form of intellectual disability. Unfortunately and invariably, those that they are older than 40, close to 95% of them, they develop Alzheimer's disease. We have a very good model for Down syndrome, which is the TS65DN, which consists of a translocation of chromosome 16 and 17, which encompass about 70–80% of the human orthologs genes, which are expressed in chromosome 21. And the beauty of it is that they have a robust learning and memory deficit and impairs plasticity. So we decided to look at this, and at the time, most of the focus was trying to identify how specific genes which are dysregulated in chromosome 21 regulate to the pathology. So we wondered whether protein homeostasis was actually altered, and we measured translation rates in the brain of those animals, and we discovered that translation rates are inhibited, and they have a feature of inhibition by the ISR, namely you have a decrease in polysomes with a concomitant increase in monosomes. Consistent with this data, when we look at pure micellation, we see that compared to the wild type, we have a decrease incorporation in the Down syndrome brain, indicating that translation rates is inhibited. We look at different ways in which, or different pathways that feed into translation initiation, EIF4F complex formation, phosphorylation of EIF4E, 40BP phosphorylation. We couldn't find any change, but we found an increase in phosphorylation of EIF2 alpha. This, we not only found it in the mouse, but we also found it in the brain of individuals who have Down syndrome. We were preparing human iPS cells from a particular patient, and we believe that this patient has a mosaic mutation because we have clones from the same patient that have 2 chromosome 21 and other chromosomes they have 3 chromosomes 21. So this is the perfect isogenic or possible isogenic control that you can use. And now when we look in those cells, the phosphorylation of EIF2 alpha is increased in the trisomic clone compared to the disomic. And consistent with this, obviously, we see a decrease in translation rates. Thus, the ISR is activated in the brain of mice and individuals who have Down syndrome. We found that the PKR branch of the ISR is activated, and if we remove PKR and we cross it with the TS65DN mice, we are able to reverse this increase in phosphorylation of EIF2 alpha. But more importantly, when we look at the behavior of the mice, this is before training. When we look at after training, the mutant animals, the Down syndrome animals, they have an impairing memory. But if we were to remove PKR genetically, or if we were to treat with an inhibitor of PKR, we are able to reverse the cognitive decline, which is associated with the disease, which we believe, at least in part, is controlled by the ISR. Now, we can study, if you wish, memory in slices, and instead of one can do a very weak training protocol, and you can see a response which decay over time, and this is measured the strength of synaptic connections. But if we were to do the same stimulation several times, now you have a response which is sustained, and this is the one which is protein synthesis mediated. When we look at that response of synaptic potentiation, in red, we find that in the mutant animals, the synaptic potentiation is impaired. But now, if we were to correct or we were to inhibit the PKR branch of the ISR, we are able to reverse those synaptic changes that we believe, or many of us, we believe that they are contributing, at least partly, to the cognitive deficits. Now, what is the nature of the mRNAs whose translation is altered? So we started those experiments like probably 6-7 years ago, and we did polysome profiling, and we isolate the RNA and we sequence it, and we have, of course, changes in the mRNA stability and transcription and translation. We obviously focus on only in translational changes. And as you can see, there's a large number of mRNA whose translation is inhibited in the Down syndrome brain. But now when we cross it with the PKR, we are able to reverse more than 70% of those changes. Now, as a field, we end up trying to look for particular mRNAs that could explain a given phenotype. Now, so we end up cherry picking and using a bias approach. But there are multiple candidates that one could potentially needs to validate, and maybe you need a combination of products. So instead of taking a particular mRNA as a response, we say maybe what we could do, instead of correcting one mRNA, maybe we can correct the entire translational program. And in order to do this, we decided to inhibit the phosphorylation of EIF2 alpha genetically, or promoting the activity of EIF2B with Israel. And when we do so, when we cross the Down syndrome animals with animals that have a decreasing phosphorylation of EIF2 alpha, Now, we correct the defects in translation, but more importantly, now animals that they have memory problems, the memory problems are gone, and the changes in plasticity the same way. And consistent with this data, when we use ISRIF, which works like a biological stapler, which puts together EIF2B, the different subunits, and promotes EIF2B function, now we found that we give it to the mice, and we are able to essentially reverse the cognitive decline in those animals, and also the changes in plasticity that are associated with it. So we have only one conclusion that was sort of unexpected at the time, and trisomy 21 leads to a proteostasis stress, which is characterized by the activation of the integrated stress response. And as you know now, when the integrated stress response is activated, you impair memory formation. And that gives us the possibility that essentially we could use perhaps ways to alleviate this. Now, the integrated stress response, I have to say, that is emerging as a main pathway in which different conditions would strike the brain, say during development or during adulthood, they converge on the activation of the integrated stress response and ultimately lead to cognitive dysfunction. And now we believe that perhaps with different inhibitors or different strategies, some of which we are working as we speak, we could essentially inhibit this so-called maladaptive feedback loop, which essentially leads to a cognitive decline. So how I am doing, Wayne, with time? You are mute.
You still have over 5 minutes left, Mauro.
Okay, perfect. So what characterizes, in my view, what gives power to the integrated stress response is specific mutations that have been identified in key factors of the integrated stress response pathway, which leads to an activation of the ISR. One of them, there are mutations in CREP. This is the phosphatase cofactor, which essentially renders the phosphatase specific to EIF2. And the other ones are mutations in EIF2S3 or EIF2, the gamma subunit, in which essentially, as a consequence, will lead in both cases to activation of the ISR and inhibition of ternary complex formation. So to demonstrate whether this mutation leads to cognitive decline, this is the mutation that you have in PPP1R115B or CREP, which is associated with severe microcephaly, short stature, and intellectual disability. And those are fibroblasts from the patients, and they have an increased activation of the ISR, as you could see here. Now, Lucas Reineke in my lab, who trained with Bill Merrick, decided to create or reproduce or introduce the same mutation that we have in humans into the mouse. And we have generated the R658C mutation. And as one would expect, either in the brain or primary fibroblasts from those animals, we have an increase in phosphorylation of EIF2 alpha. There's a decrease in translation rates as determined by either puromycylation or polysome profiling. And when we look at the memory of those animals, they have a severe memory impairment and also an impairment in this plasticity of those changes, long-lasting changes in synaptic function. More importantly, when we inhibit the ISR using genetics, now we are able to reverse the cognitive decline and those deficits in synaptic plasticity, which are associated with the disorder. The beauty of this disorder, unlike any of the models that have been used before, is that you have a persistent activation of the ISR. And this is the only thing that is happening in the brain of those animals. So as we speak, we believe that by using this model, we will be able to identify what is the sort of the ISR translational program and that we have in the brain, because in the other models that I mentioned, Down syndrome, Alzheimer, or many other models, not just the ISR is activated, but there is many other pathways which are dysregulated. So we have some data, but I am afraid that I won't be able to share it with you. But they are quite interesting indeed. So I would like to thank the people who did the work in my lab, especially in the Down syndrome paper was Ping-Jun Su, Lucas, Sean, and Sanjeev, and I'm still here in San Francisco. I'm doing my sabbatical with Peter Walter, and we've been collaborating with Peter for the last, I would say, 5 years or so. And of course, I need to thank you, Nahum. He gave me the great opportunity, gave me free range to do pretty much whatever I wanted to do in his lab. And probably I wouldn't be here if I wouldn't have, his support. And I would like to thank you. I'm happy to answer any questions that you guys might have.
That was great, Mauro. Thank you very much. I have a number of questions on the Q&A list. You can look at them as well. I may rephrase them a little bit. Bill Merrick is remembering a paper from Nahum's lab. I believe this is your paper as well, Mauro, that with the GCN2 knockout mouse that actually had reduced long-term memory. So how does that fit in with GCN2, PKR, PERK? What is the, why you get different results with different EF2 alpha kinase knockouts?
This is a great question. And this is the only piece of data that doesn't fit the entire story. So if you were to inhibit GCN2 and you were to give a weak training protocol, so in order to elicit a long-term memory in those animals, so why we call them the super smart mice, is because we give a weak training protocol such the wild type doesn't learn. If you were to do it in the GCN2 knockout, the EF2 alpha knocking, the PKR deficient, actually, if you were to give a PERK inhibitor in the brain, as Colby Rosenbluth has shown, invariably you lead to an enhanced memory and the wild type doesn't learn. When you give a normal training protocol, the PKR knockout, the EF2, and many of the other animals are completely normal. So, but the GCN2, they have a memory impairment. Why that happened, I don't know. One possibility would be that some of those kinases are phosphorylating something else beyond EF2 alpha, or you have a compensatory mechanism that is taking place in the absence of GCN2. That's the best answer that I can provide.
So from Justin LeBeau, so why in Down syndrome is PKR activated? Why is it PKR, not the other kinases?
So why it is, I don't know. So we look at the activity of the all the EF2 kinases, but HRI. And we found that PERK and GCN2 were not activated. And we discovered that PKR was activated. And as you know, now there are several papers that show that you have endogenous cellular RNAs that can form double-strand RNA structure and then activate PKR, because as you know, PKR has a double-strand RNA binding motif. In addition to this, there's a couple of papers from Nari Kim that mitochondrial RNAs essentially can activate PKR. And in those disorders like Down syndrome or Alzheimer, where the ISR is activated, where PKR is activated, now you have oxidative stress. So there's a possibility that now these RNAs will leak out from the mitochondria, get into the cytoplasm. But this is something that we are pursuing, trying to identify how this is taking place.
A question from Xiaoshang Zhu. Is it possible that increased translation of some mRNAs is in EF2 alpha, do they negatively regulate memory formation? So in other words, is the, instead of positively regulating memory formation, there's translation of some mRNAs getting rid of negative regulators?
Well,
that way memory is increased.
I make, in the introduction, of course, these are keynote lectures. I wanted to make a very simplistic approach. But not Randy Kaufman and not many of us, we have seen, if you were to even get rid of the two alleles in serine 51, right, replace it by alanine, we don't see an increase in translation. So it might be, or we believe that, and according to Dawson's paper that I just mentioned to you, it has to be translation of a specific mRNAs. So, but the same question goes when you treat with a tunicamycin or any other agent that activate the ISR, although you inhibit general translation, there are mRNAs which are more susceptible than others. And what confers that susceptibility, I think it remains to be seen. And perhaps that, if we were to identify a motif there, this could explain those which are also more susceptibly activated by inhibition of the ISR.
Okay, the next question is from Kate Gerard, from Sheffield Hallam University. And do you think it would be reasonable to look at ISR inhibitors as potential candidate treatments for neurodevelopmental disorders, or all neurodevelopmental disorders? Or is that too wide?
I think it would be too wide. Definitely, it would be too wide. And I think, all of us, we try to come out and we say, okay, will be the explanation for everything. And it might not be. I think those inhibitors will be effective when you have an activation of the ISR, when you have a deviation of homeostasis, and perhaps under certain conditions, because we know very well how the ISR works. Under certain conditions, those inhibitors could be effective. So yeah.
All right, and one last question from David Ruggiero. Which physiological conditions in the brain lead to EF2 phosphorylation? Does synaptic activation activate the kinases that phosphorylate EF2?
Actually, it's the opposite. So conditions which, as I show you in the paper from the Dawson in our work, in a learning paradigm, what you have is you have an inhibition of the ISR. Okay. Now, what we are developing now is a model in the lab in which we can sense every single stress that you can think of, in every single cell type that could activate the ISR or not. But now, if you were to have seizures or if you were to have neurodegeneration, those are conditions which, pathological conditions that lead to an activation of the ISR. For some other work from my colleague Duke Kalakos, she has found that there is different cell types in which the activation of the ISR is a very important requirement for the function of the cell. So we may have cells in which the ISR is inhibited for the function, and others in which the activation of the ISR is also important. So I think that's the beauty of this pathway, that I think it has a lot of promises for the next few years.
Great. There's a few more questions that maybe you can answer offline, Mauro. Thank you very much again for a great talk. We're going to have a 5-minute break.